So my name is Hannah, and in the P2P CPP project, we are interested in finding out ways of making a better way of reaching intracellular targets for drugs that are based on biological molecules. The project was initiated when Klaus Olesen from STIPE had some question, and he approached basic researchers. And in STIPE, what they want to do is to target an intracellular protein-protein interaction. And they had some challenges where Klaus, he approached first Ulf Simonsen from biomedicine and later also me from molecular biology and genetics. And later we have been interacting mostly with Richard Bethel. So for reaching something that's inside the cell, that's generally a challenge for any kind of drug that you want to develop. So our aim is to develop a safe and efficient means of getting novel kinds of drugs into cells. So of top pharmaceuticals by sales now, the small molecule drugs are really not don't look like they will be the major ones in the future. And for small molecules, you can get something over the membrane if they are small and hydrophobic. So they have intracellular targets, but when you have something that's larger, like an oligonucleotide, a protein or a peptide, it needs to penetrate the membrane, and typically they will not do that by themselves. So one way that has been heavily studied is to use a cell-penetrating peptide to couple to these biologics in order to get them in. The first kind of CPP that was discovered was a small part of an HIV protein that's very positively charged. It has a lot of arginines and some lysines as well. And this was shown that just attaching this short peptide to practically anything can get it into the cytoplasm of other cells. And it has been used in numerous projects, both basic research, but of course also very much to get something that interacts with a mechanism that's medically relevant to get to interfere with that mechanism inside cells. But there are still no CPP-based drugs on the market. See if I can change this. This is just a very short list of some examples of CPP-based drugs that have been in clinical trials. And it's just to give you an overview of that they are targeted against various diseases, cancer being an important one, but also many neurological diseases. And of course, there are both a lot of CPP-based drugs that have been first tried in preclinical trials and that are under development in preclinical trials, and also a lot that have gone through human trials. And the curious thing is that it's still really unknown how the CPPs can actually penetrate the membrane. So a membrane is, of course, very hydrophobic, and you have something that's very highly charged. So how is it that it mediates the passage to the cytoplasm? And you can find numerous reviews just from this year where they are lining up all these kinds of models. So in principle, you have either that it's an energy-independent direct penetration where somehow this peptide can go directly through the membrane, or that it's based on an energy-demanding endocytosis pathway where a receptor, or it can be clathrin-mediated or non-clathrin-mediated, or you can, various studies have shown any of these mechanisms to be important for the CPPs to get into cells. And it's probably true depending on the conditions, the cells you have, and the concentration, for example, of the CPP and what it's coupled to, that all of these mechanisms can be in play. But to say overall, it's just still not understood. And one challenge that we have is that these are typically used, and of course, the first thing you do is to do a cell culture. And CPPs are very well tolerated by cells when you have them in culture. You can at least have tens of micromolar concentrations and not see any apoptosis of the cells. But the picture can be somewhat different when you start to inject them into, and of course you normally start with mice. So various CPP-based drugs have been reported to be toxic, and the reason for the toxicity is generally unknown. It's not predicted by the in vitro assays. So we have been setting up some alternative where we're not just using a cellular assay, but some alternative in vitro assay to determine what are the effects of the CPPs. And importantly, we have been aiming at having something that's physiologically relevant, both in regards of concentration and timescales. So it has to be something that's quick, and it has to be something where we are at maximally 1 or 2 micromolar and see an effect. So what they do in Ulf Simonson's lab is to measure vasodilation in rat arteries. So they can do that for all kinds of drugs. If you have the normal artery here and you add something, you can measure if it affects the arteries directly. And what we do is electrophysiology. So the experiments are performed by Shengya Zhang in my lab and Stefano in Ulf's lab. And one thing that we see is that the cargo that's attached to what we have studied is the quintessential CPP, or the first discovered, namely TAT, that the cargo also has a large influence of the toxicity, the cardiovascular toxicity, and the effects on the electrophysiology of these cell-penetrating peptides. We have tested TAT alone, of course, a TAT-conjugated peptide that has been through a phase 3 trial. It was not successful there, probably because of drug-drug interactions. It's a potential drug for acute ischemic stroke, and it's called NA1. It's just conjugated to a small peptide. And then we have another TAT-conjugated peptide it's very loosely based on an initial STIPE peptide, and we call this ODIN. And what we see is the electrophysiology and the in vitro dilation effects are, there's quite a good agreement between the results we get in one assay and the other. But when we start to inject them in vivo, we see that they can be toxic in different manners. So in our in vitro assays, we see that TAT and ODIN both have some stronger effects than NA1, but we see that NA1 is actually probably the most toxic in vivo. But the toxicity for that one is, our hypothesis right now is that it's histamine related, whereas it's a more cardiovascular effect that we see for the TAT and the ODIN peptides. So the other thing that we should address here was how is this an open science project and how is it affecting us? So I tried to look up what open science is, and you can find all kinds of definitions according to Wikipedia. So I have divided this into, this is just the introduction from Wikipedia, and it has to do with how you disseminate to all levels of society, and how you engage and make it easy to access the data that you're generating. So these are marked here in blue, and our aim to fulfill this is to present at international conferences. We talked some about how we will share our data. Our aim right now is to upload at bioRxiv and then link to that by, for example, the ODIN portals. Of course, publish open access, and when we have when we can, then do popular public communication about it. And another aspect is that it should be within the collaborative networks, we should be transparent and accessible. And just within our network, we meet very regularly, and of course we exchange ideas and especially peptides, because we are all studying the same peptides, and we have journal clubs. So we just, if we find something, we will discuss it very often on mail with the others in the network. And apart from that, we also all of the participants here have various networks where we discuss both formally and informally and present at our department meetings or when we meet colleagues. So about the data sharing, this was an example from Twitter from the recent SFN conference, where they're talking about advantages and disadvantages there might be about this very open data sharing. So I'll just give an example here of data that I have where I'm challenged, and I think it's very interesting and important to an interesting and important aspect of the open science. So if you see in the two rows here, these are different treatments that we have on the y-axis, the signal, and on the x-axis, the time. And it's not that you need to understand at all what the graphs are showing, but on the 3 repeats over here, I think you can appreciate that it's very reproducible. If we wanted to, we can easily normalize the first signal and the second signal and the third signal and get a number and put all of these into a nice graph. Whereas the data that we receive over here, well, again, what we see is reproducible, but the signals are much harder to interpret and to get a nice single number from it, because the signals fluctuate, and importantly, it's not reversible. So we don't come back, which is what we really like to do in our electrophysiology measurements, is to say that we have something that was not influenced by something else, because we can wash the signal away. So for this part here, it's pretty straightforward to make a figure and to present and share the data. It will be somewhat harder to do it here and to analyze. And another part here would be, how can we make sure that data that is maybe more difficult to explain is also published? And I think when you read a paper, you always see that they have a nice hypothesis, and then they do 4 experiments or have 4 figures that all support it. And then they can tie a nice bow around it and say that this was the result. But I'm sure that for many of these articles, there will also be experiments hidden away that didn't support the hypothesis. But for science to go forward and for others to also learn, others who are working on similar problems, it could be very beneficial also if you shared data that you have that do not necessarily directly support your hypothesis. So in the next year of our studies, we will we are finishing the first part, and then after Christmas, we will include more biophysics with Daniel Utzon's lab. And again, our goal is to develop a better understanding in vitro of the molecular mechanism and how we can do something, how we can have assays that better predict in vivo toxicity.